Well, it's really fortunate for me to be able to follow Lucinda in that she's set up a lot of the information about climate change. I want to talk about how climate change might be affecting mercury. I shouldn't really claim to know how it is affecting because it's very complicated. In fact, that's one of my messages about how the mercury cycle is very complicated, and I'll need to educate you about mercury in order to discuss the possible effects of climate change on mercury. And here's a good example of how complicated mercury is. And in fact, that's part of the message, that it's just a complicated thing. But the thing I want you to get out of this diagram is that virtually all the mercury that gets into lakes and contaminates fish, and fish contamination is the problem, comes from the atmosphere. About 1/3 of the mercury is natural, and 2/3 is the result of human emissions from the intentional use of mercury in products and mostly coal combustion. So it's delivered over the lake and the watershed by the atmosphere, and it washes out in wet deposition, and also it just settles out in dry deposition, both to the lake surface and to the watershed. And in particular, I want you to notice that it's landing in this wetland over here on the right, and there's this word next to that wetland, methylation. And I'm going to talk a lot about mercury methylation in this talk. It's a critical step in the contamination of fish, in that down here at the bottom, where in the middle of the lake where the fish are accumulating mercury, it's only MEHG that's accumulating in the fish. HG is the chemical symbol for mercury, and MEHG is how a lot of scientists abbreviate methylmercury. It's not actually an official abbreviation, but it's a shorthand. So the methylmercury is only produced in anoxic environments by bacteria. So one anoxic environment is that wetland, and the other one that's pretty likely is the sediment at the bottom of the lake. And this is the methylmercury molecule. Now, I thought I'd show it to you because I'm going to be talking about it so much. The big sphere is the mercury, and it's attached to what's called a methyl group. It's a carbon in the center, the dark atom, and then 3 hydrogens. The methyl group comes from the same root as methane. On the left is a methane molecule. It's just a carbon atom with 4 hydrogens. Methylmercury is very similar. It's a carbon atom with 3 hydrogens and that 1 mercury atom. And as I said, it's the only form that accumulates in fish. And only a portion of the mercury that lands in an aquatic ecosystem is methylated. So this methylation step is critical. It's really controlling how much mercury is available to get into the fish. It's a simple organic compound, that 1 carbon atom. Chemists refer to any molecule that has carbon in it as an organic compound, so this is organic. It behaves much differently than inorganic mercury. Inorganic mercury, which is not connected to the carbon, will not bioaccumulate in any animal. And the reason the methylmercury does is a very odd accident of the environment. It attaches to an amino acid, and then it's built into protein. So it's in the protein of the fish, and that's the muscle. That's what we like to eat, the fillet. Other contaminants often accumulate in the fat of fish, but methylmercury is not in the fat. It's in the protein. Now, the inspiration for this talk really came from an observation that my colleague Bruce Monson made at the Pollution Control Agency. He's responsible for analyzing all the fish data that the DNR and the health department collect. And so he was looking at the fish data, and we have a whole lot of fish data in Minnesota. He looked at the data that started being collected in 1982 up through 2006. In that time period, lakes had been sampled 1,707 times, including about 845 lakes. So essentially, these were lakes that had been sampled twice, looking for time trends in the contamination of fish. So here's the distribution of the lakes across the state. There's a lot in the northeast, in the northern lakes and forests, that green area, but there's also many outside that green area. And what he found was that the mercury in the fish was going down over that whole time period about 1% a year. And that was very good news, and it made total sense to us because we'd been working very hard to reduce mercury emissions to the atmosphere. We put controls on incinerators. We removed mercury from latex paint. We took mercury out of alkaline batteries. You've probably noticed you can't buy a mercury fever thermometer anymore. That's a state law. We've done a lot of work to reduce mercury emissions, so this made sense to us. That was the good news. The bad news is that Bruce is a very good scientist, and he started looking at the data more carefully, and he found that actually the real data had been declining even faster up into the mid-1990s. This is the heavy red line. Then there was an inflection point, and then it started increasing. And he used a statistical package that could look at nonlinear data. So the linear line was the best fit if you only used a straight line, but if you allowed the statistical package to bend and turn, there was a much better fit if there was a decline than an increase. So there was actually about a 4% per year decline up until the mid-90s, and now we've been increasing fish contamination about 1.5% per year since the mid-90s. And Bruce is not the only person that's observed this. This increase since the mid-90s has been observed in several of the Great Lakes and in southern Wisconsin and the small lakes. Now, to explain this and talk about it, I'm going to be drawing on information that was developed by mercury scientists working in 2 locations in Minnesota. Of course, we're down in the red spot down near Mankato, but there's 2 places that mercury scientists work in the north. Just north of Grand Rapids, there's the Marcell Experimental Forest run by the U.S. Forest Service, which hydrologists and many other scientists have been working there pretty much in obscurity since the 1960s. And they've been doing very good work, and a lot of mercury research has been performed there. And the other place I'll be talking about is Voyageurs National Park, which is a wonderful place to visit and to work, and has several large lakes, but also has a peninsula that has about 30 small lakes on it that are quite isolated from human contamination, and a number of mercury scientists have sampled those small lakes, and there's some interesting results there. Now, I told you mercury was complicated. This is a computer model of how mercury behaves at a small lake in up at the Marcell Experimental Forest. This model was developed by Neil Hines for his PhD thesis in environmental engineering. And of course, how complicated it is part of my message to you, but it's not that bad, really. The deposition from the atmosphere is coming in at the top of the model, and the fish contamination levels are coming out the right-hand side. And there's lots of other details, but what's interesting is that a EPA scientist named Paul Koka a few years ago ran a number of these different models. There's actually about 4 or 5 different mercury models much like this. And he ran them, and what he found was that it all simplifies down to that fish are contaminated in direct proportion to how fast the mercury is coming down from the atmosphere, if you hold all the other variables constant. So here on this graph, we have mercury in fish on the vertical axis and the variance in atmospheric mercury deposition from that contamination of air pollution coming down. And it's a direct proportional thing. It's a straight line relationship. That's what the models predict. And that's good news. That makes sense that we wanted to reduce how much air pollution there was of mercury, thinking that it would benefit the fish. And this is the prediction, that if we have half as much mercury coming down, the fish will be half as contaminated. Notice it goes through 0. If there's no mercury in the atmosphere, then there'll be 0 mercury in the fish. However, as I noted, about 1/3 of the mercury in the atmosphere is natural, so we can never get to 0. Now, not all the lakes have the same amount of mercury in the fish. Turns out that different lakes have different straight line relationships. And for instance, the atmospheric deposition rate is about 12.5 micrograms per square meter per year. Anyway, 12.5 there on the, if we go out on the x-axis here to 12.5, we can see that different lakes will have vastly different mercury levels in the fish, where Lake A is a lake that produces a lot of methylmercury out of that mercury that is deposited, the inorganic mercury that's deposited. And Lake C is a lake that's really bad at making methylmercury, so the fish aren't very contaminated. And what we assume in that mercury model is that this level of efficiency is pretty constant in a given lake. So a given lake will be either good at methylating mercury, that's Lake A, and so the fish will be really contaminated, or bad, that's Lake C, or kind of medium, that's Lake B. So if this model's correct, what Bruce observed with the fish getting more contaminated since the 1990s would predict that there's more mercury coming into the lakes. Well, here's the record of mercury deposition at 5 monitoring sites in Minnesota since 1995. And it's pretty noisy data, but if anything, it's going down. So something else is going on. Why are the fish getting more contaminated if there's no more mercury? Well, the model was assuming that everything was constant except for mercury. Well, maybe that's a bad assumption. We can't assume that all the other factors are remaining unchanged. So what else could have changed so that mercury is increasing in the fish? One easy thing is that the food chain could be longer, because this is a biomagnification phenomenon. The more steps in the food chain, the more mercury will be in the top predator. It starts out at a low concentration. In fact, mercury is at an incredibly low concentration in lake water, maybe around 1 or 2 nanograms per liter. And then only a part of that is turned into methylmercury, maybe ranging from 2 to 20% is methylated. And then by the time that methylmercury gets into the top predator fish, like a walleye or a bass or a northern pike, it's 10 million times more concentrated. So that's the main problem with methylmercury, is that it's in the protein, and protein's valuable, so the fish retain it. When a fish eats a smaller fish, it keeps all the protein it can, and it's by accident keeping the methylmercury. And it gets concentrated to an incredible degree, an average of about 10 million times. So if there's an extra step in the food chain, that'll raise the concentration in the top fish. Well, we have no reason to think that since the mid-90s, the food chains have gotten longer. I mean, this could happen. Exotic animals can be introduced, for instance, but I don't think it's a widespread phenomenon across Minnesota yet. For instance, there is a Bithotrephes. It's a small animal that's gotten into Lake Superior and some other lakes from the Baltic, from the ships coming into Duluth Harbor. But it is not widespread across Minnesota yet, so it doesn't explain the higher mercury in the fish. Other explanations would be the higher average temperature that Lucinda Johnson was just talking about. If there's a higher temperature in the water, the fish will have a higher metabolic rate. They'll eat more. Fish, when they eat, they burn through the calories and they keep the mercury. So the more a fish eats, the higher the mercury concentration. So that's one explanation. one possible explanation. Lucinda Johnson also talked about the longer ice-free season. Remember, it's bacteria that are turning mercury that comes down from the atmosphere into the methylmercury that gets into the fish. And these bacteria presumably are more active when it's warmer. Incredibly, nobody's done that experiment and taken some mud and incubated mercury at different temperatures and to see if it's higher at higher temperatures. That experiment ought to be done, but it hasn't been done yet. And it's not clear what would happen because there's also other bacteria that demethylate mercury. So remember, mercury's complicated. Then there could just be a greater average methylation efficiency in lakes. But what could cause that? And that's the main aspect of what I want to talk about here today. What could cause greater methylation efficiency in Minnesota lakes? Could have increased bacterial activity due to the increased temperature, or, and here's my topic, increased water level fluctuations. And Lucinda talked about greater storm intensity interspersed with dry periods. I mean, we may not have too much different total precipitation, but if it arrives in bigger batches and greater storms, then there's going to be drier periods. And if the environment alternates dry periods and big storms, then the water levels are going to go up and down in lakes and wetlands. And the IPCC reports have also predicted more variability in precipitation, more summer droughts, and greater storm intensity. I think this is a pretty valid prediction, and it's been observed to some degree. Well, why would this change mercury methylation? Well, it's kind of a complicated story, but that's why I'm here. The bacteria that methylate mercury are SRBs, and that SRB stands for sulfate-reducing bacteria. These are bacteria that take sulfate when oxygen is not available. So in an organic-rich place like the bottom of a lake or a wetland, that organic matter is available for bacteria as energy, and the bacteria will eat that, or the bacteria will eat the organic matter. And if there's water on top of the organic matter, such as there often is in a wetland or a lake, that water inhibits the penetration of oxygen. Oxygen can't make its way through water very fast. So the bacteria will use up the oxygen, and when the oxygen's used up, other bacteria take over and respire sulfate instead of respiring oxygen. And this is a fascinating phenomenon. So really, we're going to talk about two molecules, sulfate and methylmercury. You've already met methylmercury. Sulfate is a sulfur atom surrounded by 4 oxygens. And what the sulfate-reducing bacteria do is they eat the carbohydrate, and they respire the sulfate, and there's a little arrow there, and they produce some carbon dioxide, some hydrogen sulfide, H2S, that's that bad, rotten egg smelling gas, and a little bit of water. So sulfate-reducing bacteria make hydrogen sulfide, and they happen to turn inorganic mercury into methylmercury at the same time, and nobody knows why they do that. It's maybe some accident of metabolism. We don't think that the mercury is high enough concentration for them to be detoxifying it, but maybe that's a mechanism. Anyway, they happen, the more that the bacteria respire sulfate, the more methylmercury is made. So I already talked about that the sulfate-reducing bacteria are active when there's low oxygen. It's sort of the opposite of acid mine drainage, to bring in something else you might have heard of. Acid mine drainage is the opposite of sulfate reduction. It takes reduced sulfur, in this case pyrite, which is the FeS2, and acid mine drainage can occur without bacteria. It just is a chemical reaction. The pyrite reacts with oxygen, a little bit of water, and it makes rust, which is the ferric hydroxide, the iron hydroxide, and sulfate, and acid. So I thought I would bring in this acid mine drainage concept because, for one thing, I want you to be worried about the sulfate rather than the acid. Because the sulfate has, for many years, been regarded as a benign ion that doesn't hurt anything. It's not toxic, not very toxic anyway. It's about as toxic as salt would be, sodium chloride. So let's ignore that acid part a little bit. So then on the bottom half of this slide, I'm repeating what I told you before, but I'm doing it backwards at first. The hydrogen sulfide that the sulfate-reducing bacteria make can oxidize on its own, just like acid mine drainage. So H2S plus oxygen makes sulfate and a little bit of acid. And then on the bottom of the slide here, it's reversible by the SRBs, the sulfate-reducing bacteria. In this case, the equation is going from the right to left. The sulfate and a little bit of acid combine to make hydrogen sulfide and oxygen. So let's put this in a picture in a wetland. There's water on top of organic sediment. The bacteria want to eat that sediment. And in fact, the reason that organic muck builds up in a wetland is because the water's there that keeps oxygen out. And so when there's not much oxygen, the organic matter doesn't degrade very fast and it can build up. So the bacteria run out of oxygen and they start using sulfate instead of oxygen. And they make H2S, and that H2S can actually make pyrite again, which is that component of acid mine drainage. And as you can see, it does produce a little bit of methylmercury, MEHG. So that's what happens when there's water on top of sediment, either in a wetland or in the bottom of a lake. And after a while, the bacteria will run out of sulfate, and the methylation will stop. But then what happens in a fluctuating precipitation environment that we've been discussing? The water evaporates, the sediment is exposed to oxygen, to the air, and that chemical reaction occurs. It's just like acid mine drainage, where the H2S and the pyrite chemically form sulfate. You don't even need bacteria for this. And then the sulfate's just sitting there. It needs a little bit of water, but the sediment's damp. And the SRBs, the sulfate-reducing bacteria, are just dormant. They're sitting down there waiting for the water to come back. And then get a thunderstorm, late summer, and the water comes back. And the whole thing repeats again. We make more methylmercury. So there's a lot of mercury sitting in the sediment because only a small proportion is methylated. And if we cycle the sulfur back and forth between sulfide, sulfate, with increased water level fluctuations, we make more methylmercury. So this is my hypothesis that this is a possible explanation for this increase in fish contamination. It's not that there's more mercury. In fact, probably there's less mercury coming down from the atmosphere, but there's a lot of mercury stored in soils because of thousands of years of atmospheric deposition. And it's sitting there and it's available for methylation if the bacteria methylate it. Now that's all conjecture and speculation, but there is some data to support this. I want to point out that about 10 years ago, an enterprising young scientist named Jeff Jeremiason, who just introduced me, was in charge of a project to artificially add sulfate to a wetland to see what happened. And here's Jeff, about 10 years ago, building a synthetic rain machine up at the Marcell experimental forest. And I was there and I helped spray sulfate on the wetland. And then Jeff was the first author of a quite influential paper that showed that adding sulfate to a wetland does produce methylmercury. So this is adding extra sulfate, but it proves the point. Now this was not the first time that this had been shown. It had been done in small experimental plots before by Canadians and some Swedes, but this was the first time that half of a wetland had been sprayed. Several acres of wetland was sprayed with sulfate. And then after that experiment, the project was taken over by Jill Coleman Wasik, who began her PhD research in 2005 at Marcell. And she was going to continue following these experimental additions of sulfate to see what happened. And then she had a terrible natural disaster occurred. The summer after she started, in 2006, there was a major drought at Marcell. It was the lowest water level in that wetland that had been observed in the 40 years that they'd been monitoring it. So she thought all was lost. This is terrible. But she's turned it into her PhD thesis because it dried out the wetland. And the next year when the water came back and flooded it, the sulfate concentrations were very high because the sulfide had been converted back to sulfate, and the methylmercury levels were much higher as a result. So that turned out to be a great natural experiment, but she didn't know that when she was first getting into it. Now, I'm not going to show you any of the data because it's complicated, plus she hasn't finished her thesis yet, and you'll have to watch for that. So my conclusions out of all this is that we don't really know what factors are causing the mercury to increase in fish. The water level fluctuations and associated climate change are a possible explanation, but there's other viable hypotheses, including just the temperature increase and perhaps even longer food chains. Who knows? Plus, it could be that multiple things are happening simultaneously. Maybe they're all happening. Maybe mercury's going down and temperature's going up and water fluctuations are happening. And I've characterized the increase in fish as though it's happening everywhere. Actually, a little over 60% of the lakes are increasing. And it's statistically significant, but other lakes are doing different things. So different lakes do different things, we know that. And these unresolved questions are being addressed as scientists are able to obtain funds and support. And I urge you to keep track of the research results. Thank you.